So now we'll discuss some acquired causes of hemolytic anemia. So the acquired causes of hemolytic anemia is autoimmune hemolytic anemia, microangiopathic hemolytic anemia, infections such as malaria that we will discuss in infectious disease chapter. And another one is paroxysmal nocturnal hemoglobinuria. So here we'll only discuss autoimmune hemolytic anemia and microangiopathic hemolytic anemias. Infection we'll discuss in infectious disease chapter, and paroxysmal nocturnal hemoglobinuria is a rare disorder. It will not usually be asked in the Plab 1 exam, so we will not discuss it here. So the acquired cause of hemolytic anemia, the first one is autoimmune hemolytic anemia. What is autoimmune hemolytic anemia? Autoimmune hemolytic anemia is an anemia due to autoimmune reaction in which antibodies are formed against the RBCs, which will result in antibodies are formed against the RBCs, which will result in hemolysis. So there are two types of autoimmune hemolytic anemia. One is a warm antibody induced autoimmune hemolytic anemia, and the other one is cold hemagglutinin disease. They are named warm antibody and cold antibody because the antibodies that are formed in both of them, they react with the RBCs at different temperature. The antibodies in cold hemagglutinin disease, they are IgM category, and they react with the RBCs on a temperature at lower temperatures, at temperatures that are lower than the body temperatures. They cause agglutination of the RBCs and hemolysis of the RBCs. So that's why it is known as cold hemagglutinin disease. And the antibodies that are formed in warm antibody hemolysis, they react with the RBCs at body temperatures, that is 37 degrees centigrade. So that's why it is called warm antibody autoimmune hemolytic anemia. And the causes of formation of warm antibodies, that is mostly IgG. It's idiopathic. Mostly it is idiopathic. There is no known cause, but it can occur secondary to autoimmune diseases such as SLE, lymphoproliferative diseases such as lymphoma and CLL. So lymphoma and SLE and CLL, they are associated with autoimmune hemolytic anemias, the warm type. And cold antibody autoimmune hemolytic anemia, the first one is Microplasma pneumonia. We discussed in the respiratory chapter that Mycoplasma pneumonia is associated with erythema multiforme as well as autoimmune hemolytic anemia, the cold type. So cold antibody autoimmune hemolytic anemia is idiopathic mostly, but it can be associated with Mycoplasma pneumonia infection and infectious mononucleosis. So Mycoplasma pneumonia, infectious mononucleosis, cold antibody autoimmune hemolytic anemia, SLE, lymphoma and CLL, warm antibody hemolytic anemia. The presentation or presenting symptoms will be same as of hemolytic anemia, that is anemia, increased reticulocyte count, spherocytes on a peripheral blood film. As we discussed along with the hereditary spherocytosis, the only way to differentiate between autoimmune hemolytic anemia and hereditary spherocytosis is through a COMBS test. So if the COMBS test is positive, then it will be autoimmune hemolytic anemia. And if it's associated with Mycoplasma infection or infectious mononucleosis, then it will be cold or If it's associated with the SLE, lymphoma, and CLL, then it will be warm antibody autoimmune hemolytic anemia. So a COMBS test is used to differentiate between autoimmune hemolytic anemia and hereditary spherocytosis. Now, some points about COMBS test. There are two types of COMBS test. One is direct COMBS test and one is indirect COMBS test. The difference between these two types of COMBS test is that direct COMBS test detects the antibodies that are attached to the surface of RBCs. So direct COMBS test or direct antiglobulin test detects the antibodies that are present on the surface of RBCs, and indirect COMBS test, they detect the antibodies that are present in the serum. So direct COMBS test is done on the RBCs and indirect COMBS test is done on the serum. Direct COMBS test is used in the conditions in which antibodies are formed against the RBCs or in which antibodies attacked on the RBCs. What are the examples of those conditions? The example of those conditions include autoimmune hemolytic anemia, RHD disease of the newborn, and ABO hemolytic disease. These are some examples which can be detected by a direct COMBS test. While indirect COMBS test is done in two circumstances. One is it is done for blood grouping and cross matching. What do we do in a direct COMBS test? In indirect COMBS test, we take the recipient serum. We are going to do this test for a blood grouping and cross matching. So we'll take the recipient blood and we'll add a recipient, add a donor's blood sample in this serum. So if this donor's blood, if these antibodies are against or does not match, or if there is mismatch, what will happen? That these antibodies will combine with the antigens present on the surface of RBCs. So this is the first step of indirect COMBS test. The recipient serum is taken and it is mixed with the donor RBCs. And if there's a mismatch, what will happen? These antibodies, for example, if the person blood group is A and the recipient blood group is B, so these will be anti-A antibodies and they will target the red blood cell of the donor. Now, in the second step of this indirect COMBS test, we'll add COMBS antibodies, and these COMBS antibodies will clamp the RBCs, will cause agglutination of the RBCs. So we'll say that indirect COMBS test is positive if there is agglutination. And if there is no agglutination, then it means this blood can be transfused because it is a negative indirect COMBS test. So this is how indirect COMBS test is done for blood grouping and cross matching before doing a blood transfusion. And the second circumstance in which we do this indirect COMBS test is a circumstance in which we want to detect. If you people can remember from gynae chapter that a female with a negative blood group, we need to check her anti-D antibodies level. So the first use of indirect COMBS test is in cross matching for blood transfusion, and the second is to detect antibodies, anti-D antibodies that are usually IgG that can cross the placenta and enter into the fetal circulation and can cause hemolysis. So in this scenario, we will also do indirect home test to detect antibodies in the mother serum. While direct home test is directly done on the patient RBCs, and a positive direct home test means there are antibodies present on the surface of RBCs, which are present in uh, autoimmune hemolytic diseases or ABO blood group mismatch or RD mismatch. So this was a small detail about direct home test and indirect home test and autoimmune hemolytic anemia. I hope it's clear, everyone. If there is any question, then you people can ask. No questions?